I'm presenting the findings from the CD19 TPAL study, which is a pan-European study of CD19 CAR T cell therapy in patients relapsing post-allogeneic transplantation with B lineage ALL. And this was a CD19 CAR study which utilised virus-specific T cells. So these were T cells that were primarily responsive to EBV, and then we selected those and used them as a population to transduce in a first-generation CD19 CAR. So we transduced with a retrovirus, so more derived from HIV, if you like, to transduce T cells, but the T cells were responsive against the EBV virus. So it's a common problem following transplantation, and if you can boost immunity to that, you can provide an advantage to the patient when they're vulnerable to viral reactivation, particularly with EBV post-transplant. So that's why we chose a virus-specific T cell population. But those cells were then transduced with a retrovirus to express the CD19 CAR. But of course, that virus is disabled, so it cannot infect the T cells and doesn't have an oncogenic potential in the future. This was an attempt to reduce toxicity by using a T cell population that would be capable of responding to viral reactivation, and that the viral reactivation would provide a stimulus for proliferation and expansion and memory formation. Uniquely, there was a vaccination strategy that was incorporated into the second cohort of the study to try and improve persistence, because we found that in the first cohort, CAR T cells alone did not persist when administered alone. So the second cohort received vaccination, and we showed that vaccination improved the persistence of the CAR T cells. Overall responses at 1 month were seen, but the longer term efficacy was limited, unfortunately. So this was a unique strategy which hasn't been employed before, and it may be that there are ways that we can improve that. But ultimately, going forward, we are recommending the use of a second generation CD19 CAR, which is what we're using in our follow-up study. It's a very exciting new field, and there are significant toxicities associated with the sorts of approaches that have been discussed and have been widely reported in the press. And so really the challenge going forward is firstly in rolling it out to larger patient populations, and secondly in trying to reduce the toxicity. And the CD19 TPAL study was trying to provide a more physiological stimulation rather than the sort of souped up immune activation that you see with second generation CD19 CARs, but it appears that the latter approach is probably going to be more successful in the longer term.